So what is pH? It is the negative logarithm of hydrogen ion concentration, or it is the logarithm of the reciprocal of hydrogen ion concentration. And this is the formula shows here. Buffer. It is a mixture of weak acids with their salts of strong bases and resists sudden change in hydrogen ion concentration even after the addition of hydrogen or hydroxyl ion. Now Henderson-Hasselbalch equation is also shown here. pH equal to pK plus log salt divided by acid. Here some example of buffer pairs. Can see that. Now state the significance of buffer in the body. Acids produced in the body as a byproduct of metabolism tend to decrease the pH of different fluids like intracellular fluid, interstitial fluid, blood, etc. But the pH remains steady due to the presence of buffer. Now osmosis and osmotic pressure is also described here. Osmosis is defined as the spontaneous flow of water. Osmosis is defined as the spontaneous flow of water from a dilute solution to a more concentrated solution. So when they are separated by a semi-permeable membrane. Now the difference between osmosis and oncotic pressure is that the osmotic pressure will be greater if the molecular weight of a substance is low. Name the factors regulating osmotic pressure like ADH, which is antidiuretic hormone, aldosterone, which is a mineralocorticoid, the third factor. State the Van 't Hoff's law of osmotic pressure. The osmotic pressure of a solution varies directly with the concentration of the solute in the solution. What is the clinical importance of osmosis? The clinical importance of osmosis is the osmotic force is the injection of hypertonic solution of magnesium sulfate to reduce the volume of the brain or lower the pressure of the CSF. Other different importances. What is dialysis and its clinical importance? So here it is. It is the process of separating crystalloid from colloid by diffusion through a membrane adopting osmotic force. Okay. Now surface tension. The force with which the surface molecules of a liquid are held together is called surface tension. Some substances that increase surface tension and some decrease surface tension. Here it is the Gibbs-Thomson principle in relation to surface tension. The substances that lower the surface tension become concentrated at the interface. Discuss the clinical importance of surface tension. Surface tension is involved in the process of digestion, and bile salts use this criteria for the absorption of lipids. The inward force generated by alveolar surface tension increases diffusion of fluids into alveoli from alveolar capillaries. Okay. In postnatal life, type 2 alveolar epithelial cell in lungs secrete a surfactant called DPL, which prevent collapse of lung alveoli in experience. So in TCA cycle, name the enzyme, coenzyme and inhibitors are used. So these are the enzymes. Coenzymes and inhibitors are arsenite, dietary deficiency of thiamine. Why it is called citric acid cycle or TCA cycle? Since the citric acid first formed, why is citric acid cycle said to be amphibolic in nature? Citric acid cycle provides metabolites for anabolic processes such as fatty acid and amino acid synthesis and gluconeogenesis. It also provides CO2, H2O and ATP. Okay. So where and under what condition? Show the number of ATP formed in TCA cycle. So this is pyruvate to acetyl-CoA 3 ATP, isocitrate to oxalosuccinate 3, alpha-ketoglutarate to succinyl-CoA 3, succinyl-CoA to succinate 1, succinate to fumarate 2 and malate to oxaloacetate 3. So total 15. One molecule of pyruvic acid being oxidized in the TCA cycle yield 15 ATP in one molecule. So the colloids are the substances which do not diffuse through parchment or animal membrane. The size of each colloid particle is 10 to 100 nanometer. What are the method of determining the size? By ultrafiltration, diffusion rate, ultracentrifuge, etc. What are suspensoid and emulsoid? Suspensoids particles carry a definite electric charge. They are easily precipitated if the charge is neutralized. Emulsoid are very stable and not easily precipitated by salts. All colloids of the living cell exist as emulsoid. Okay? They are hydrophilic. An emulsoid may be changed to suspended by dehydration and then is precipitated. They carry electric charges also. Now gels, they are quasi-solids. High pressure is required to squeeze water out of a gel. Formaldehyde can remove water from the gel and hence it is used in the histological process of fixing tissue. Now imbibition and the protective colloids are also written here. So saturated fatty acids. These fatty acids have no double bonds in their structure. For example, acetic acid, propionic acid, etc. Name the monounsaturated fatty acids. The examples are palmitoleic acid and oleic acid. Name the polysaturated fatty acids like linoleic acid, linolenic acid. Now discuss the clinical importance of essential fatty acid, EFA. So lipids of gonads contain high concentration of polyunsaturated fatty acid. Okay, prostaglandins are synthesized from arachidonic acid. Leukotrienes are also formed from arachidonic acid. Express the biomedical importance of bile. Now we know that bile is secreted from liver, synthesized in liver and stored in gallbladder. And bile is very important in the absorption of fat. Cholic acids permit the absorption of many valuable insoluble